Okay, I will present some results on our research on drugs of abuse and designer drugs. My name is Helander. I'm working at the Karolinska Institutet, the Department of Laboratory Medicine, and I'm also at the Karolinska University Laboratory here in Huddinge. And what is a designer drug? It's a substance, a psychoactive substance that has been chemically designed to circumvent legislative actions. So this means it's essentially legal until it's classified. Just an example, here you see the chemical structure of amphetamine, which is one of the most abused drugs in Sweden. And if you simply change the structure by adding a methyl group in this position, you have 2-methyl amphetamine. And this is a narcotic substance, but as of today it's legal to use and to buy. So it has not yet been classified. So this is an example of a designer drug. There are also other names for this kind of substances. Internet drugs, because they are sold usually over the internet. Legal highs, because they are legal until they are classified. Spice is used for synthetic cannabinoids, smart drugs, and so on. So there are a lot of different names. They are also sold over the internet as masked products, for example, as research chemicals that are only used for research, but they are usually abused as bath salt or as plant food. But today the common denomination of these kind of substances is new psychoactive substances, or MPS in short. So MPS is the name I will use in my presentation. And before these substances are classified, they can be sold openly on the internet as legal narcotics, which of course is a bit strange, but this is the legal effect of the narcotics classification in Sweden. And the reason why people use designer drugs or MPS is that, of course, they're not illegal, so people can buy and use them without being prosecuted. Some also believe this means that they are less dangerous, but it's usually the opposite. You buy them on the internet, like any other things. It's easy, safe, probably, but cheap. They cost less than standard illicit drugs, for example. A lot of people are curious about these substances. They're called psychonauts. They want to do travels to see what effects they get of the new substances. And the one important thing is also that they are usually not detected in standard drug testing, so it means that people can continue using drugs of abuse without being detected. And the European Monitoring Center for Drugs and Drug Addiction and Europol, each year they present statistics of the new substances that have been detected in Europe each year. And here you can see the results from 2005 through 2015. And the bars present the new substances detected each year. So each substance will only be covered 1 year. So this means that, for example, in 2010 there were 41 new substances, and in 2015 there were close to 100 new substances detected in Europe. And the different types of substances is presented. You can see it's the standard class of drugs of abuse, like opioids, benzodiazepines, cathinones, tryptamines, and synthetic cannabinoids. And the increase started in 2009, and that was based on the discovery in the end of 2008 that some of these smoking herbals that you could buy in shops, in gas stations and so on, contain synthetic cannabinoids. And before that, people saw that the people taking these substances, they behaved like if they have been smoking marijuana, but they did not test positive for cannabis. And that was the reason, because it was not cannabis as such, it was synthetic cannabinoids. And since 2010, there have been more than 500 such substances detected in Europe. So the number of drugs of abuse today is enormous. Last year I was in US presenting our work on opioids, and I then stated that when this was detected, these kind of new substances, it was the end of the recreational drugs world as we knew it, and it also means a lot of problems for us that are working in the detection of drugs of abuse. So it was a new era. And when all these drugs are freely available, there you can buy them open on the internet, this also increases the problems. And I can give you some experience from the legalization of cannabis in Colorado that was effective from January 2014. And of course, when cannabis became legal, the use increased. You can see here some statistics that the increase went up about 45%, and Colorado is the state with the highest cannabis use in the US. You can also see that the problems increased, traffic fatalities increased, at least doubled. You can see that traffic deaths went up 150% before and after the legalization. And you can also see that public health worsened. The visited at emergency departments because of cannabis increased 52%, and all hospitalizations went up 150% about. It's also interesting to note that the excess of cannabis after legalization has increased much. 1 year back in Colorado, there were almost 500 retail stores selling cannabis, and this is higher than the number of Starbucks retails and the number of McDonald's. So it's easy to get access to cannabis today in Colorado. The results I will present here are mainly from our results in Sweden, and it covers the result from a project called the Strida project that we started in 2010. And the focus of this work was to check the availability and trends of these new psychoactive substances in the country, and also to try to identify harmful substances and in analytically confirmed cases of intoxication. And the design of this work was as such that it was a collaboration between the Karolinska and the Swedish Poisons Information Center, which is a 24/7 service for clinicians and the public in case there were questions about drugs of abuse, about intoxications, to get treatment advice and so on. And all emergency departments and intensive care units in Sweden were invited to take part in this work. And in case there was suspected intoxication by a new MPS, they contact the Poison Information Center to get treatment guidelines. And then they were instructed to send samples to us for analysis, when we usually receive blood and urine samples, but sometimes also drug materials that were handed over by the patients or relatives and so on. We performed the analysis, and we reported this back to the clinicians that were responsible for the patient. The Poisons Information at the same time collected clinical information from medical records, and all this together we put together and reviewed in each case. And sometimes we also had to do a reanalysis of the samples because the substances that were suspected were not yet covered by our analytical methods. And all the time we added up our knowledge about new psychoactive substances, we updated the analytical methods and so on. So this is the design of the study. And the number of cases in this presentation, I covered the results from 2010 through 2015. And you can see in the first year we had about 100 intoxication cases, but the peak was in 2014, and then we had close to 800. That is more than 2 acute intoxications by these drugs per day. And then we know that this figure is about less than half of all cases, because only half of the consultations with the Poisons Information Center also resulted in blood and urine samples that were sent to us. So the real number was at least twofold higher. And the samples and cases that we received were from all over the country, from the far north to the far south. And the number of cases were received approximately in proportional to the number of inhabitants in Sweden. So most people from Svealand and Götaland. And the patients were aged from 8 to 71 years, and most people were aged 15 to 30 or 35, which is common for people abusing drugs. The youngest persons were usually smoking synthetic cannabinoids, and the older people were usually taking stimulants like amphetamine variants and so on. Actually, we had one patient that was a newborn, and the samples were received from this newborn, and the mother had taken these drugs, and the father was a dealer of these drugs. So actually, the age range should be from 0 to 71. And about 75% of the cases were from males. Much of the focus was on analytical problems, because we only included analytically confirmed cases, and I'm working at the Karolinska University laboratory. And the reason we used analytically confirmed cases is that it's hard to get reliable information just by asking people about if they use drugs, and denial and underreporting is very common. So for that reason we collected blood and urine samples to check for the actual use, previous use of drugs. And drug testing is usually done in a two-step variant. It starts with preliminary testing. It's an immunoassay screening based on antibodies, but antibodies are specific or less specific, and usually what you get is an unspecific result. It means that it can be the substance you are interested in. It could also be a similar drug or some completely different drug with the same epitope. So although results are quick and rather inexpensive, they are unspecific, because there is always a risk of false positives and false negatives. And this can be done both in the lab using automated systems, but it could also be as a kind of point of care testing close to the patient with dipsticks and so on, which are even less reliable than the laboratory methods. To obtain true result, you need to do a confirmatory analysis, and this is done by chromatography combined with mass spectrometry. And we mainly use liquid chromatography combined with mass spectrometry. This method is of course more time consuming and more expensive, but the result that you get is reliable, it's qualitative and quantitative. It means we can tell exactly what substance the patients have been taking, and also the concentration, which will indicate the approximate time since intake. So this is the two-step process. Problems related to analysis of new psychoactive substances is that usually drug testing focuses only on the top 5, which are in Sweden cannabis, amphetamines, cocaine, opiates, and benzodiazepines. Sometimes we also cover a few more like LSD and ecstasy and so on, but usually it's only the top 5 that are covered. So it means the new psychoactive substances are never included in routine drug testing. So this means that we have to think differently, we have to behave differently, we have to include new methods to also cover the new substances. And this is a debate that I published in the Swedish Medical Journal a couple of years ago. And the main complication is that if you test for more substances, it will also increase the cost, and that is the reason why some testing is not performed. It is true that these substances are structurally similar to the classical illicit drugs, and this means that some immunoassay testing targeting the classical immunoassay drugs will also detect these, or at least the assays will react. But unless you have a confirmatory method that covers also the new drugs, they will not be detected. And we have examined this in studies for opioids, for benzodiazepines, and so on, and it's the same. We can also see that some substances are certainly not detected in the immunoassays, like for these designer fentanyls, where you can see that this form methoxybutyrylfentanyl is not detected at all, and this is because of steric hindrance. When you add a chemical substituent to the basic molecule, you will also hinder the antibody to attach to the epitope, so it will not be detected. Another problem related to these substances is that many of these are very closely related. They may have the same or almost identical structure. There are isomers and so on, so it means that the mass will be more or less the same. And also the chromatographic performance will sometimes be the same. For example, in liquid chromatography, they will have more or less the same retention times. So this also increases the problems related to testing for drugs, new drugs of use. Sometimes then we need to have very long analytical times to be able to separate them. In this, it's methoxy PCP, which is a dissociative drug, and you can see where we have the methoxy group in 3 or 4 position. It's a sterical isomer, and it's really hard to separate unless we use a very long retention time. So this means we had to think in a different way when we analyze these substances. And when you combine a two-dimensional analysis, that is liquid chromatography separation and then mass spectrometry detection, you can of course improve the liquid chromatography. You can also improve the mass spectrometry, and that was the choice we had to make to improve the analysis and routine detection of new psychoactive substances. So we started using high resolution mass spec. We used the Q-Exactive Orbitrap system, and which has been very effective, and the reason that we have been able to analyze all these substances. And we have long list with new substances where we add the information that we get. We can run these methods both in single MS or in tandem MS mode, and for some substances we really need to do the tandem MS, which increases the specificity. But for some it's sufficient to use single MS. And this is due to the high resolution of the mass spectrometry. Just an example, sometimes we don't have access to reference materials, and then we have to look at the mass spec and see what could it be. Here you have the mass spec for fentanyl, which with the target ions of 105 and 188, and these are typical for fentanyls. And we know the structure of fentanyl, but also know that the fragments will be like this, and the head gives this 216 fragment. If we have a new fentanyl, which is not yet known, we can compare with that of fentanyl, and in this case you see that we see the 105 and 188, but instead of the 216 we see 214, so this means that we have lost 2 hydrogens. And this means also that we get a double bond, so this is acryl fentanyl instead of fentanyl. So this is one way that we can detect new substances even before they have been clearly identified and before reference materials is available. And it's really important to do this to confirm what substance is used. This is an example of MDPV, which is methylene deoxypyrovalerone, which is a substance that has been used a lot since 2012. And in blue you see the consultations with the Poisons Information Center, and in orange you see the analytically confirmed cases in the STRIDA project. And you can see that the consultations remained high even in 2015, but from mid-2014 we did not receive any positive results in our analysis. So even though people contacted the Poison Information Center about the suspected MDPV intoxication, we could not confirm that this was the substance. So instead it turned out to be a very similar compound called alpha PVP. And you can see that there were a lot of positive results for alpha PVP during this time, but there were no consultations. So the patients or the people that were taking these substances, they believed they took MDPV, but it was actually alpha PVP. And after that, there have been a lot of very similar, structurally similar substances that are based on MDPV or alpha PVP, and we have identified these in our research and published a lot of data on this. We have also done a lot of metabolomics studies because we want to see how these substances are metabolized. In some cases, the metabolites are more potent or more harmful than the parent substances. And we can also use these metabolites as target analytes in urine drug testing, for example. And this has been done for example, for fentanyls, where we decided not only to analyze the parent compound, but also one metabolite. And if we see the metabolite, we know that the substance has been used and has been metabolized in the body. So a common standard metabolite for all fentanyls is the norfentanyl metabolites. We also know that, like the illicit drugs, there are a lot of harmful effects of the new substances because they are very alike the classical illicit drugs. The most recent statistics in Sweden says that about 350 people have died from these substances in the last 3 years, and these are cases where these substances have been specified as the cause of death. In our work from 2010 through 2015, we have identified at least 3,000 severe intoxications by these substances. And as I mentioned before, that figure should at least be doubled because not all cases are included in our STRIDA project. And some of these substances have been identified as especially harmful. These are some results of an opioid called MT45. It was a candidate prescription drug in Japan, but it was not approved for use in humans because of adverse effects, side effects. And then sometime identified this substance in some publication, started to synthesize it and sell it as a new psychoactive substances. And this caused a lot of problems. The first adverse effects that we noticed were hearing problems, and people lost instantly after one dose of this substance. They did not hear, and in some cases this was permanent. And later on, when we examined other cases, we identified severe skin and hair symptoms, dermatitis, folliculitis. People lost their hair, and even if they were dark-haired, when the hair came back it was all white. And some also lost sight, they were blind, they got cataract and required surgery to get back their sight. So that was really harmful substances. And as I mentioned, we also received a lot of drug products from these intoxication cases, and we had all in all about 250 different drug products, and they could look very differently. In some cases they were sold like research chemicals, you could see the structure and so on. In other cases they were completely unlabeled, so we did not know what it was, so we had to do analytical investigations. And we have summarized this in a publication. And for example, this product called benzofury was sold first with a content of 5 APB, then as 6 APB, and then with a substance called 5-IT. And this 5-IT is extremely harmful, and when it was introduced as a designer drug, in just a couple of months, 14 people in Sweden died because of this substance. Another example is MDMA or ecstasy. That is usually sold in tablets like this, but it could be PMMA, which is a structurally related substance instead of MDMA. And there is no way you can tell this just by looking at these tablets. So when you take such a pill, you never know what is the true content. And these are variants of methamphetamine, which is a commonly abused drug. And this is the chemical structure of methamphetamine. And here you see the corresponding structures of MDMA, that is ecstasy, and this other drug, PMMA. It's also usually called a Superman drug because of this Superman logo on the tablet. And you see that these three substances are very similar in structure, but they have completely different clinical effects, and especially MDMA is much more harmful. Also, a lot of fentanyls have been sold, and fentanyls, like opioids, are very harmful substances because the side effects of opioids are that you lose consciousness and you stop breathing, and then if you don't get into hospital or get an antidote, you will soon die. And these substances were sold in nasal spray flask without any labeling, so you did not know what they contain. One example is also that many of these fentanyls were sold as fake drugs. For example, Prince, he died 2 years ago, and he had in possession standard prescription drug with a marking exactly as the prescription drug. But when they did the analysis of this tablet, it was a fake tablet that contained fentanyl. So Prince died from an overdose of fentanyl. In the project, we have also been able to follow the effect of legislation of these substances. And here you can see the results for some MPS opioids over time from 2012 through 2016. And you can see in green were cases where we detected the substance or got consultations to the Poisons Information Center. And the red vertical lines are when they were classified in Sweden, and then they were stopped, marketed openly on the internet, but then they were replaced by other substances. So first there was this AH7921, and when it was classified in 2013, it was replaced by this MT45, as I told you about. And when this was classified, started the introduction of fentanyls, and so it has continued till today. And there are a lot of different substances that can be introduced as legal alternatives. It was also presented in a publication recently that this shows that actually when you classify substances, there will be a demand for new legal ones to be introduced. This can, so to say, drive the market. And there are thousands of different variants that can be imagined. For example, in these two books that were published in the 1990s, more than 1,000 different substances are described, how to synthesize them, and what the effects are. You can also find this information in scientific publications and in patent applications and so on. So there are thousands of different substances that are just waiting to be used or introduced as designer drugs. So to sum up, the results from our work is that the focus of our project on acute intoxication cases from emergency departments and intensive care units was very successful. The collaboration between the laboratory and the Poisons Information Center was also successful because we were able to introduce acute intoxications from all over Sweden, and by this we also identified the most harmful substances. Other experience were that to be able to perform these results, you need real-time information about which novel drugs are introduced and sold. This means checking home pages for MPS vendors, need to check drug chat forums and so on to see which are the substances in use. And of course, we gained a lot from the analytical examination of the products that we received, because these were the products that were used at the time of the intoxication. And all the time we had to update our analytical methods. So this means that we had to add on all the new substances that we identify. And as I told you before, since 2010, the number is above 500. So this means a lot of problems, really. And this is just a list of the substances that have been reported in Europe so far in 2018. And you can see we are up to 46 just this 1 week back. And this adds on all the time. So the final number for 2018 will be at least one new psychoactive drug introduced per week in average. And what about the future? We don't know, but as long as substances are classified one by one, it will take some time, and there will be some time that the new substance can be sold openly without risk being prosecuted or without risk going to jail. So we don't know what will happen, but certainly there will be more new drugs introduced. Thank you.